Hey class, in this recording we're going to talk about membrane proteins. As we're looking at the proteins that are embedded within the plasma membrane, membrane proteins make up a minority of the number of molecules, so about 2% of the molecules, but about half the weight, the overall mass of our membrane. And as we look at these membrane proteins, they can be broken up into categories. We have integral proteins, and by integral proteins we mean that they penetrate all the way through the membrane. They go from the extracellular side to the intracellular side. As we look at these transmembrane proteins, that's another name for them, in that they are going to be transmembrane. They go all the way through. They cross from one to the other. And as we're looking at the membrane proteins, they have hydrophobic and hydrophilic regions. The hydrophilic regions are going to be in contact with water-containing solutions like the cytosol of the cytoplasm and the extracellular fluid. And the hydrophobic regions are going to be embedded within the plasma membrane. And these membranes can either drift around freely within the membrane, others can be anchored to the cytoskeleton to keep them stationary relative to the entire cell. We also have peripheral proteins within our plasma membrane. These peripheral proteins are going to be either extracellular or intracellular, one or the other. They don't go all the way across the membrane. And these are typically going to be tethered to the cytoskeleton and help anchor it in place. So here we can see our transmembrane proteins going all the way across. Sometimes they can just be a skinny little protein. Sometimes they can be quite large and can vary in size dramatically. As we're looking at membrane proteins, these membrane proteins have a variety of functions. These functions include being a chemical receptor or message receptor. They can act as a secondary messenger system. They can act as a channel that allow chemicals to go in and out. They can act as an enzyme to speed up the rate of chemical reactions. Or they can act as a cell identity marker. They can help to identify the surface of our cell with a carbohydrate tag so that we can tell one cell apart from another cell. Or they can serve as cell adhesion molecules. They can anchor one cell to an adjacent cell. As we're looking at these transmembrane proteins, the receptors, proteins, are going to bind chemical signals. They will take the signal from the extracellular part of the cell and receive that signal. Usually, they will have a conformational shift, or they'll have a slight change in their physical shape when they bind to that chemical signal. We can have secondary messenger systems within the cell that are based on membrane proteins. So the receptor on the surface of the cell receives the signal, and then they communicate to other proteins within the cell to initiate chemical reactions inside of the cell. These internal chemical reactions are called secondary messenger systems. The enzymes that are present on the cell are going to speed up chemical reactions or catalyze chemical reactions. A classic example is the brush border enzymes within the small intestine. These enzymes are embedded within the plasma membrane of the microvilli and allow for the cell to digest chemically digest food as the food is flowing through the small intestine. As we look at channel proteins are going to allow hydrophilic solutes and water to pass through the cell membrane. Recall, our cell membrane is a phospholipid bilayer. It has 2 layers of phospholipids. And this phospholipid bilayer has a hydrophobic core with its fatty acid tails. So for water or hydrophilic chemical compounds to go in and out of the cell, there needs to be some kind of a protein that facilitates transport so that those molecules aren't exposed to the fatty acid tails. Some channel proteins are always opened. Some can open and close or are gated. We have ligand-gated protein channels. Sometimes those are referred to as chemically gated protein channels because they respond to chemical messengers, also known as a ligand. Sometimes we can have voltage-gated chemical channels that respond to a change in the voltage of the membrane, or mechanically gated that respond to physical stress being placed on the cell. As we're looking at the carrier proteins, those carrier proteins will bind a solute, and think back to our chapter 2 discussion. A solute is something that's been dissolved in the solvent. So it can bind a solute and move a solute across the membrane. Usually these are going to be hydrophilic solutes that need help getting across the membrane. Some of them are going to be pumps, and when we say pumps, they're going to move a chemical solute against its concentration gradient. Because they're working against the gradient, they are going to consume cellular energy, ATP. We can have proteins that function as cell identity markers. The cell identity marker proteins have a glycoprotein on the surface, or are a glycoprotein with a glycogen subunit exposed on the surface. These act as identification tags, and it's a way for our body to differentiate a red blood cell from a white blood cell, from a muscle cell, from a bone cell. We can tell the different cell types apart with these glycoproteins. And then we can have cell adhesion molecules. Cell adhesion molecules are going to mechanically link one cell to an adjacent cell to help give structural stability to the tissue of our bodies. We also can have proteins that work as secondary messengers. These secondary messenger proteins are oftentimes referred to as G-coupled proteins. And when we look at these G proteins are going to take an extracellular signal. So here's that extracellular signal represented by that red molecule, and a receptor will bind to it. And when that receptor has the extracellular molecule bind to it, it's going to release a G protein intracellularly. And the G protein will travel to an adjacent protein within the cell membrane known as adenylate cyclase. And this adenylate cyclase will then, once the G protein has attached to it, take ATP and turn the ATP into a signaling molecule called cyclic AMP. Cyclic AMP is going to be our secondary messenger, and that secondary messenger will then take an inactive enzyme, an inactive kinase specifically, and activate it. And once we activate the enzyme, we now have an activated kinase within the cell. That activated kinase can then go and activate other enzymes by phosphorylating them, adding an inorganic phosphate group to them. So the activated kinase will take an inorganic phosphate group and then use it to activate other enzymes, and those other enzymes will then have various metabolic effects. They can activate other chemical reactions. When we think of secondary messengers, cyclic adenosine monophosphate, cyclic AMP, or CAMP for short, and calcium 2-positive ions are the most common secondary messengers. It's also worth emphasizing that over half of our drugs that we use within Western medicine are based on G-coupled protein cascades. That's all we have for this recording on proteins of the plasma membrane. If you have any questions or comments, please feel free to post them on the class discussion board. And as always, happy studies.